Okay, very good morning to all of you. So today we are observing our lecture number 18, uh, and this is the first last lecture of the hydroponics. Right? In last lecture we have seen the aeroponics. So in aeroponics there are 2 points, 2 to 3 points: nutrient solutions, commercial aspect, and the advancement. So how commercially it is used in the market. So already, as in relation to nutrient solutions, there are the points simple, but we have to consider how the nutrient, that is fertilizers, are used. So already we are knowing the inorganic ions generally obtaining from these soluble salts of the fertilizers are there, and they are essential for the higher plants, as there are some organic compounds such as iron chelates may be present. So it is firstly reported by scientist Steiner in 1968. The physiological role of these elements and in the plant life cycle, it is having the essential. It is also shown by Tej and Jigar in 1998. There are currently 17 elements are considered for the most of the plants. And these are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, iron, copper, zinc, manganese, molybdenum, boron, chlorine, and the nickel. So, as per the hydroponics technique, Salisbury, a standard methodology for the plant biological research, it is given. Second, scientist Ross in 1990 was shown, with the exception of carbon and oxygen, which are supplied to the from the atmosphere, and these are essential elements which are obtained from the growth media. Second part, in relation to nutrient solution hydroponic system, as aqueous solution generally in the water. So mainly elements such as sodium, silicon, vanadium, selenium, cobalt, aluminum, and iodine are given, and others are considered beneficial because some of them can stimulate the growth. Then there are some compensate the toxic effects are there. So elements or they may be replace essential nutrients. It is shown by Trejo in 2007 and other scientists. The most basic nutrient solutions considered in the composition as in nitrogen, phosphorus, and the potassium, along with calcium, magnesium, and the sulfur. So these are the supplemented micronutrients are there, and they generally maintain the nutrient composition for electrical conductivity and the osmotic potential of the solution. Right. So already we are knowing osmosis mechanism. So therefore, from higher flow of solids from higher concentration to lower concentration. So these different parameters in the nutrient solution, they give the details as the pH. It is a one parameter which helps for the acidic alkalinity condition of the solution. This value generally it is having, these are the pH is nothing but the free ions of hydrogen and OH group present in the solution from 0 to 14, and therefore pH effect on the availability of the nutrient to the plants. Right. Similarly, pH it is also a composition which having the elemental speciation and the bioavailability to the plants are there. The speciation indicate the distribution of elements are there. So how it is to be given, like our we are taking the injection, so for the particular disease, so it goes to the this part. So like this one, pH plays the important role here. The process of the these chemical and physical forms, as in the form of free ion soluble complexes, chelates, ion pairs, and the solid and gaseous phases are in different oxidation state. It is shown by scientists in 1998. The important feature of nutrient solution is that they must contain the ions in the solution and in chemical form, and that can be absorbed by the plant. So in hydroponic system, the plant productivity is closely related with to the nutrient uptake and the pH regulation, and it is firstly shown by the Marshner in 1995. Each nutrient show differential responses to change in pH of the nutrient solution. So pH it may be changed later on, so we have to maintain this. The in the nutrient solution, NH3 ammonia only forms a complex with hydrogen ion for a above 7 pH for a pH range between 2 to 7. NH3 is complemented present as NH4, increasing pH above 7. NH4 decreases while the concentration of NH3 augments. Right. So it is also shown by scientists. Tyson is one scientist and others in 2007. They have shown to determine the nitrification rate. Response is a perlite trickling as a biofilter, that is the root growth medium we are using. So it is exposed to the hydroponic nutrient solution and it is varying to NO3. Then concentration of pH level 6.5 to 8.5, it is founded that nitrification was significant. So all experiments are done by the scientists here. The potential of increased levels of unionized ammonia, it reduces the nutrient uptake of the plant and the micronutrient precipitation takes place. It is generally seen at the pH 8.5. Phosphorus is an element which occurs in the form that are strongly depend on the environmental pH is there. So other chemical forms they are in the nutrient or water, they are to be responsible. Here the pH is slightly acidic, that is 5. So in alkaline and highly acidic solution, the concentration of phosphorus decreases. So you remember all these nutrient condition. Then in hydroponic, as it is to be supposed as a standard methodology for plant biological research, there is one authority, NAM, so and others in 1996 they have shown the different experiment as the used the ethyl either polyethylene or the stainless tube in the solution tank, and the flow type it is get the double. So this is also interesting part is there, so we have to give the attention toward this one. The having 41 70 or 81% of the cooling load of the hydroponic in the greenhouse respectively. Then Villela and other scientists in 2004 evaluated the cooling of the nutrient solution at up to the 12 degrees Celsius. Slight cooling is normally generally water temperature is 25 degrees Celsius. Then heating exchange the device productivity of the variety to the strawberry. So they have checked for the strawberry. Then nextly the deep sea water DSW can used in the preparation of nutrient solution due to the nutrient content. Likewise they are used, and cold DSW deep sea water, they are pumped inside the pipe through the cultivation bed. So these are some experiments done by the scientists. Then oxygenation of the nutrient solution. So the consumption of oxygen increases when the temperature of nutrient solution increases too. Then it produces an increase in the relative concentration of CO2 in the root. The environment, if the root aeration is not adequate, so it shows this condition. It is shown by Morard and other scientists. Then oxygen and nutrient solutions, they are generally showing depend on the crop demand, which is having the higher for the photosynthetic activity as it increases. It is also shown by scientists in 1999. A decrease below 3 or 4 mg per liter of dissolved oxygen, it inhibits the root growth and produce change to the brown color. So it is also shown, and it is due to the first symptom of the oxygen lack, and it is shown in 1983. So like this, all scientific nutrient conditions, they are to be checked by the scientist. Then competition for oxygen in the root environment. So there is a dense matting is there. So in the substrate, the oxygen diffusion generally alter, and there is a supply of the oxygen is there. So like this one, we have to consider about the nutrient solution condition. In nutrient solution, NH3 and all these one, they plays important role. So hydrogen ion generally for a pH range between 2 to 7, it is also important. Already we have seen about the role of ammonia, right? So then main part composition of the nutrient solution is there, as there was previously stated as nutrient solutions usually contain 6 essential nutrients: N, P, S, potassium, calcium, and the magnesium. So thereby Steiner created the concept of ionic mutual ratio, which is based on the mutual ratio of anions of NO3, H2PO4, and SO42. The mutual ratio cations K plus calcium, magnesium is important. Such a relationship is not just about the total amount of the solution, which keeps that the ions together, and therefore relationship them take place, and the performance can be negatively affected. So it is shown in 1961 and 68. In this way, the ionic balance constraint make it impossible to supply one ion without this, introducing a counter ion, and a change in the concentration of one ion must be accompanied. So I will send the PDF of this lecture today, so you read it properly again and go through it. There are some good examples in spinach seedling. Temperature of irrigation water 24, 26, and 28. Then it is evaluated for 8 weeks. Right. So therefore, leaf length, leaf number, and total fresh and the dry biomass. Then per plant, it is observed. So plants grown at elevated temperature with optimum growth, and which are recorded for 28 degrees Celsius. In tomato plant, the rate of water nutrient uptake by roots it is changed. Then solar radiation and temperature is at 35 degree Celsius. So short day and long day plants are like this. It is to be studied for the short and the long term water and nutrient condition. So these are some parts are there in the as a nutrient medium. There is having the increased fruit dry matters are there. So in the particular lycopene in the tomato, so it is also studied by the scientists. So what is the effect later on in the fruits? So in tomato it is studied. Secondly, there is having the study done by the Juraj in 2006. So certain nutrient solutions, they under special environmental conditions, they shows the relative proportion and shows the proper concentration. Then the plant nutrient uptake, generally it shows the analysis of the leaves done by the Hoopt scientist in 2002. So different examples are there. Then Graves in 1983 observed that the temperature below 22, it is dissolved oxygen in the nutrient solution, is sufficient to cover the demand of the elements in tomato plant. Then secondly, as there are certain physiological processes are there, so temperature 22 degrees Celsius, so oxygen demand is not covered by the nutrient. So like this one, below and the above temperature, it is important part is so that the temperature has a direct relationship to the amount of the oxygen consumed by the plants and the reverse relationship. So here you observe the photographs of the tomato and the other nutrients. So they are to be studied as per the different levels of concentration ranges, essential mineral nutrients according to the various authors are there. So all these records considered for our study. So thanks to all. So here today we are completing our credit first, and the credit second already we have completed, landscape and the gardening. So now we are starting our third credit, post harvest technology. Okay, have a nice day.